Good morning, good afternoon, and good evening. My name is Stratos Davlos, and I am the CTO at Innoplexus. Innoplexus is a small bioinformatics startup with offices in India, Germany, and the United States. So thank you very much for joining today's technical talk. We are very pleased to share with you the work we've done in generating novel molecular structures with deep neural networks. It is not a secret the cost of developing drugs is very, very high, and it's unfortunate because it doubles every 9 years. This is also known as the E-Rooms law. So if you had $1 billion in 1970, you can potentially produce 10 drugs. And with the same amount of money, inflation adjusted, you can barely make a single drug today. So there is a lot of data out there, and if you look at the scientific data proliferation, it follows an exponential curve. Just the last 2 years, we had 90% of that data generated. At the same time, the ability for pharma to take advantage of the data produced remains linear, and the gap increases on an ongoing basis. In computing, as you know, we enjoy Moore's law, where the compute power doubles every 2 years. And the question is, how can we take advantage of the data that's available plus the compute power and bandwidth and change the ability to generate drugs at a faster and lower cost pace? So the only way to do this is with AI, and our startup is in the business of leveraging AI to accelerate drug discovery. Today we'll talk specifically about small molecules. Small molecules are very important. They are important in the development of new therapeutic agents. They're also important because you can use them to probe a biological function. They can inhibit a specific function of a protein, and then can also be used to disrupt any protein-to-protein interaction. To create a new molecule, it costs approximately $260 million, and it's about 3 years worth of effort. A small molecule needs to satisfy several physiochemical and biological criteria so that it's a good candidate to meet the requirements for being a drug for human consumption. So there is a need for a solution to scan the chemical space and enable this rapid generation of new hypotheses of a viable molecular structure that is qualified to be a drug. So today we'll share with you a deep learning technique that we use to generate novel molecules, and that was done on NVIDIA hardware. And we took up the molecules that we generated through the same stringent tests and requirements that handcrafted molecule would go through to qualify for the subsequent stages. All right, so at the very high level, we acquire a lot of data. We apply structure to it, so we extract various entities, apply structure, push it through some biological filtering, and eventually use the data sets to train a neural network, a variational auto-encoder that is paired with reinforced learning. Then take that structure is capable of generating millions of molecules and take it through a process where we come up with a very short list of maybe top 5 molecules that pass all their requirements to become drugs. So let's begin by talking about the data preparation. So data is the backbone of any machine learning model. For this work, we used a variety of molecular and biological data sets that we very carefully selected to include known molecular processing molecules that are possessing the desirable properties. So we used molecules that are drug-like, they're easily synthesizable, and they bind to the target protein of interest. We also included a negative data set of molecules that possess undesirable characteristics, and there are very good reasons for that, so we get into details in a minute. So the data sets that we obtained came from several publicly available sources like Zing and ChemBL, as well as unstructured information that appear in scientific literature. And we also tapped into data that we've been accumulating over the last 5 years at Innoplexus that allow us to improve the quality of the molecules that we generate. In particular, we used about 2 million synthesizable drug-like molecules we obtained from the Zinc database. And during training, this set allowed our model to learn the latent laws, if you like, of medical chemistry, drug likeness, so that the molecule would generate conforming with the rules of nature. Because we expect our model to generate specific molecular structures that inhibit a protein so that we can cure a condition, we needed to include some additional data sets so that we can orient our model towards this objective. Specifically for COVID-19, we selected the viral protease protein of novel coronavirus called 3CL-Pro as the protein target of interest. The activity of this protein is essential for viral replication, and thus inhibiting its activity would stop the viral multiplication and spread in the host. Therefore, the expectation from our model is to generate molecular structures that can inhibit the activity of 3CL-Pro. And in our training set, we included all the known 3CL-Pro inhibitors obtained by ChemBL. In order to allow our model to generalize, we also included a set of input molecules that are known to inhibit other proteins, but belong to the same protease class as the 3CL-Pro. And while we were at it, we also included molecules that inhibit proteins of the hydrolysase superclass of enzymes, as well as molecules that are known to inhibit other classes of enzymes, so that the model can learn how to eliminate molecular structures with such characteristics. The effect of training data selection ensured that the molecular structures generated inhibiting 3CL-Pro and ensuring that it does not kind of cross-react, if you like, with other proteins in the body. Last but not least, we also used a data set of patented molecular structures that came from, I think it's called ChemBL. And the reason we did this, we wanted to make sure that the molecules we generated were novel and we can further patent them if they were proven to be have a therapeutic value. So we specifically used the Variational auto-Encoder for this work. There are various generative models that we could have used, but most of them have limitations, in particular when you try to use them in an ensemble with reinforced learning. So we used a variational auto-encoder for this purpose so that we can use it with TTLP. TTLP is the tensor trained induced learnable prior. And the combination of the two, VAE with TTLP, was used to encode smiles in other proteins of molecules on the latent space. This learnable prior can be used to efficiently compute the conditional and marginal probabilities. And with this distribution, we can compute the likelihood of an unlabeled data set to belong to the distribution by marginalizing out the unobserved conditions. During the generation, what happens is we sample from the latent space in the middle using the conditional probabilities given the observed conditions from the input data sets. So as I mentioned before, the molecules are represented using smiles, the smiles format from the input data set along with their properties. So we added properties like QED and SA and PIC50 and a binary feature that indicates whether the molecule passes any medicinal chemistry filters, the MCFs. So we will discuss all of that in more detail in the next slide. Some of the advantages of variational auto-encoders are that they require less data compared to other generative models. In our case, this was very crucial because we had a relatively small data set of molecules that inhibit the different classes of proteins. As I mentioned earlier, the best thing about VAEs is that they learn both a generative model and an inference model using TTLP, which help us use it alongside with reinforcement learning. And it also learns the underlying data distribution using unsupervised learning and generates a continuous latent distribution. So let's dive in a little bit more into detail on the data preparation side. So the molecular structures from the input data sets are expressed in SMILES notation for all the downstream processing. In addition to the molecular structures, we use the chemical, biological, and drug-like properties of each molecule in the data sets to construct our training set. So this combination allows the model to learn the relationships between molecules. More concretely, we use the PIC50 values of the molecules of the target protein. The PIC50 is the negative log of IC50, which is the half maximal inhibitory concentration, which in essence is a measure of how potent a molecule is in inhibiting the activity of a protein. MCF refers to the medicinal chemistry filters. These filters check the presence of any structural alerts. For example, chemical moieties bearing any unstable or reactive groups is of an input molecule. That is a filter that is very important. QED refers to the quantitative estimate of drug likeness of the molecules. In a quantitative metric for assessing whether a molecule is drug-like is computed on the molecular properties such as the molecular weight, log P, the number of H-bond donors and acceptors, the number of rotatable bonds, and things along these lines. Finally, SA refers to the synthetic accessibility of the molecule, and this is an estimate of how easy or difficult it is to chemically synthesize a molecule in a lab environment. So if the range is between 1 and 10, 1 is very easy, 10 is very difficult, so anything that's above 6 is basically throwaway molecules for us. Another thing is that we had to create our own tokenizer to generate tokens. The goal was to convert smiles, which are really alphanumeric strings, to a representation that machine learning can use, which is really numbers, and then be able to take the output, which is numbers, and convert it back into smiles. And it's worth noting that our tokenizer maintains all the information of smiles, including, aromaticity and stereochemistry and atoms and rings and all that stuff. The QED and synthetic accessibility are computed using the RD-Kit, which is an open source toolkit in chemoinformatics. And the MCF is computed using smarts patterns representing structural alerts obtained by Surchem builds. All right, let's click on the next slide. This slide here encapsulates everything that we're doing in our model, so you can easily visualize it. So we have an encoder-decoder pair that's built on PyTorch and also on reinforced learning network. At first, we train the variational auto-encoder with a learnable prior distribution to encode smiles and map them onto the latent space. So the VAE learns the latent attributes of a molecule from the structural and experimental properties we provided for each molecule. And as a result, it constructs the latent space with each dimension able to give the conditional probability of the sample to be the desired molecule. Next, after this step, we freeze the encoder-decoder networks, and we train the learnable prior with reinforced learning to optimize the molecules with a reward function. So the reward function, and we'll get a little bit more into detail in a subsequent slide, uses 3 self-organizing maps that help to train the learnable prior in how to sample and improve sample from certain regions of the latent space, so the molecules are better, the sampled molecules are better than others. So in essence, the variational auto-encoder model learns to generate molecules conforming to the laws of medicinal chemistry and drug likeness, and the reinforcement learning component helps in directing that generation of molecules towards the protein target of interest, 3CL Pro for COVID-19. All right, a little bit about the learning objectives. So as elaborated previously, we need to learn the rules of nature, both chemical and structural. We need to learn a lot more about the binding capabilities of the molecule and its characteristics of its target protein. We need to learn what's desirable and what's undesirable molecule based on their affinity score, atomic properties, and empirical rules. And we also need to learn how to optimize those structures towards the properties that we're looking for. So let's dive a little bit more into technical details. This diagram here represents our encoder. The encoder sometimes is referred to as the recognition network of the variational autoencoder, and it is responsible for generating a probability distribution of the latent attributes corresponding to the input data set. So this probability distribution of many latent attributes is also referred as the latent space, where each dimension corresponds to a certain property learned by the encoder. Using this multidimensional latent space, we can control the properties of the molecules generated. So for the encoder, we use the combination of gated recurrent units and linear layers. The reason we use GRUs is because we wanted to make sure that, as we encode smiles are long sequences of data. So that's why we needed GRUs. We could have used other technologies attention-based models, but for this particular machinery, GRUs were used. On the decoder side, the decoder, or sometimes referred to as a kind of generation network of the VAE, is responsible for decoding the smiles from this kind of random sample drawn from the latent space. For any sampling of the latent distributions, we're expecting the decoder model to be able to accurately reconstruct the input, which is really the learning objective of an encoder-decoder system. So in other words, the values which are close to one another in the latent space should correspond to very similar reconstructions. For the decoder, we have used diluted convolutional networks to capture the relationship between non-adjacent atoms and functional groups. And this is because with small representations, the two adjacent atoms can be far away from each other. And this arrangement or relationship, if you like, can be captured using a variable diluted convolutional net. So in other words, the sequence of diluted covnets with some kind of various amounts of dilution, which is why you see the architecture of the hyperparameters you see in this slide. As I mentioned before, it was very important for us to use reinforced learning. So the selected molecules, the ability to select good molecules improves based on a reward system. So in reinforcement learning, we provide a reward to the model for good molecules so they can improve and generate even better molecules. For scoring the newly generated molecules, we use self-organizing maps. We abbreviate those as SOMs. And those were generated using the Morgan fingerprint of the molecules. So a slightly different way to represent them, so that you know the Morgan fingerprint, or sometimes called circular fingerprint, can be calculated for any valid molecule and is good at encoding the structural characteristics of the molecule. A self-organizing map is basically reduces dimensionality. In our case, we used a two-dimensional representation of the input data set, where the byproduct of that is similar molecules with similar properties are clustered together. So we developed 3 SOMs from our input data sets, which together help in ranking the generated molecules based on the likelihood to possess some key characteristics, in particular for COVID-19, inhibiting the 3CL protein, and also it's important to us that the molecules that we create are novel, so that we can further patent them if they are selected for becoming a drug. So more concretely, the ensemble of these 3 SOMs are used in the reward function are the following. So we have a general protease SOM, which distinguishes between molecules which are active and inactive against proteases, the class of the 3CL protein target. We have a 3CL Pro specific SOM, which helps in selecting generated molecules that are potentially 3CL Pro inhibitors, as opposed to any other protease inhibiting molecules. And then we have a third SOM, which is a trending SOM, and that's the one that's responsible for assessing the novelty of the generated molecule with respect to all the patented chemical structures. In Parallel to the generation of molecules based on their drug-related characteristics and rules of nature, we decided to explore whether we can do better by generating molecules based on their structural elements. And the intent really is, if that works out well, to add a reward to our adversarial network that takes into account the geometry of the molecular structures. So this was an important enhancement to our model, and the goal is to learn from these three-dimensional characteristics of the protein and its functional binding site, as well as the 3D molecular structure, to ensure that the generated molecules are optimized for fitting into the protein cavity. So first we had to decide how to represent the three-dimensional information of these biomolecules. We captured the physiochemical and structural information in matrices or tensors, if you like. And for that purpose, we used voxel representations of the proteins, of the target protein, as well as the molecule itself. Those are called ligands as far as the molecules are concerned. So in essence, we treat the protein or a ligand molecule as a three-dimensional image. For this three-dimensional image, we have 3D pixels. These are the voxels, and their value depends on the pharmacophoric properties of the atoms present in the voxel. We also have channels which represent different groups and types of atoms. So for each, so the value of each voxel is determined by the corresponding atom type and the distance between their neighboring atoms and its center. So as you see on the slide, the image on the left shows the voxel representation of our target protein, which is 3CL Pro. And the goal is to develop a model that can generate a voxel representation of the ligands, the small molecules, that are complementary to the target protein 3CL Pro in both the shape as well as the pharmacophoric properties. This can be viewed as a three-dimensional shape of the target protein and generating three-dimensional shapes that are complementary to the binding site of the target protein. In addition, we also developed another model that takes in the voxel representation of the molecules, in other words, the ligand three-dimensional shapes, and reverses them back into smiles, so generate smiles, so we can use them in our application. Let's click to the next slide. So as discussed earlier, the voxel representation of the proteins and ligands were used, and from the data perspective, this is a very large sparse matrix. Just to give you a rough idea, a single voxel representation of a protein is of a shape 8 by 24 by 24 by 24. And when you look at the corresponding ligand, the shape is 5 by 24 by 24 by 24. So when we took the DUDE data set, we have 102 target proteins and about 23,000 ligands for their respective targets. So just roughly about 225 ligands per target. Then we use the protein and ligand voxels in a 3D bicycle gun to learn the 3D properties of the proteins so that we can generate these three-dimensional shapes of ligands that can fit inside the binding cavity of the protein itself. For training the 3D bicycle gun, We used the DUD data set, and we also added from the zinc data set about 400,000 molecules. So this is a lot of data. If you get, let's say, 102 from the DUDE proteins, and you get an additional 400,000 molecules from zinc, that's a data set of 102 by 8 by 24 by 24 by 24 plus the 420,000 molecules by 5 by 24 by 24 by 24. That's almost 30 billion floating point values. And that is where we actually used and exploited rapid data frames that allow us to minimize, data transfers between the CPU and the GPU, and it was very, very efficient. So then we trained a three-dimensional captioning network for the purposes I mentioned before, to convert it back to the generated smiles. And for that network, we also used the zinc data set of about 1.9 million molecules. To handle the huge data set, we converted them into sparse matrices and stored them in a CSV file. And then we read them with Q data frames, and then used QPy to convert them back to the original matrix for training. All right, so everything we do needed to be evaluated the same way you would evaluate a handcrafted, if you like, molecule. So the molecular structures that we generated by either model went through some preliminary filtering. And for those that passed the filters, then we went through a prioritization pipeline. The filtering of the molecules was based on some common sense and practical methods. So we used the Lipinski's rule of 5, which historically is a good set of empirical rules for assessing drug likeness of a molecule. We've done toxicity and non-drug-like structure filtering, which include filtering out molecules that contain structural alerts or toxicophores. Those toxicophores, as you probably know, are chemical substructures that are correlated with undesirable properties such as toxicity. Then we also removed structurally redundant structures that were very similar, using a clustering technique. And we selected, in essence, the molecular structures that had the highest diversity. And at the moment in the lab, we are adding that to the reward system, so it doesn't have to be a subsequent step. It can be a step as part of the generation that really accelerates things. The molecules that pass our preliminary filters were then evaluated through a molecular prioritization pipeline, as I said before, for a detailed assessment. So the first and foremost property that's evaluated is the potential to generate a molecule that actually binds on the protein. We use molecular docking for that to predict the binding potential. And molecular docking is a computational method that allows the prediction of the preferred orientation of a molecule to the protein target based on the shape and other physiochemical properties. Molecular docking provides an affinity score that you've seen on the slide, and that's actually indicating the binding potential of the molecule. The lower that score, the better it is. We're actually doing significantly better than minus 7.7 at the moment. And it also details the kind of predicting binding characteristics and the molecular interactions, such as hydrogen bonding and hydrophobic interactions and things similar to that. In addition, it's also important that the molecule shows some kind of favorable admit properties. For example, we need to make sure that the molecule can be absorbed in the body through either an injection or a tablet, a pill that you swallow, an oral route. We want to make sure that once it's inside the body, it can actually distribute and reach the site of action. So we know for COVID-19 that the lungs is an area and other probably affected organs. We want to make sure that we can metabolize it and exert it, and it's important that the metabolic rate is kind of therapeutically desirable, so it stays in the body long enough, but doesn't it doesn't stay for too long. So that's important, and we want to make sure that it's safe. So we don't want anything toxic in the body or anything that has an adverse effect and by interacting to other unintended proteins or biomolecules in the body. So that is critical. In this slide, you see some results comparing the different techniques and comparing them with the molecules that were actually used to generate were used as an input. A little bit to understand the notation, VAE plus RL is the variational auto-encoder and reinforcement learning based model. 3D refers to a 3D bicycle gun, and the three-dimensional the geometric generation of molecules that fit the cavity. And you can see that the first figure shows the distribution of affinity scores for the generated molecules. And the more negative the affinity score, the better it is. So the peak that you see towards the left of the x-axis is actually desirable. So this is where we want to be. And as you can see, that the 3D model actually has much better affinity scores, and that's something understandable because the 3D model was purpose-built to kind of optimize for the binding affinity based on geometry. In terms of the log P, which is our second graph, that is indicative of the lipophilicity of the molecule, and it's important. It's an important determinant of the admit properties of the molecule. So we observe that the generated molecules lie within the desirable drug-like range of log P, which is less than 5. So that's good news. We also observed that most of the generated molecules have a synthetic accessibility less than 4, which you can see on the graph number 3. And that's really indicating that we can easily, how easy it is to chemically synthesize the molecule in a laboratory environment. So again, we have some really good results there. With respect to QED, to refresh your memory, that's the metric of drug likeness. We observed that our variational auto-encoder shows a better distribution than the input molecules, and it also outperforms our three-dimensional model. So in summary, we show that our models are able to learn the laws of medicinal chemistry and drug likeness quite well, and we're able to generate molecules that are equivalent or better than the molecules we used as an input to build those models. These are great results, and we wouldn't be able to do that without the PyTorch framework that we used almost exclusively for our work, and the NVIDIA hardware and frameworks for manipulating data sets. We used not a huge amount of CPUs. We used 8 GPUs, and those were of the V100 family of GPUs with 32 GB of RAM. And we use those in various configurations. So in the next slide, you will see the different tasks that we performed. In the number of GPUs we use, that's the light purple color, and the amount of hours those CPUs were utilized. So you can see, for example, to train the model on the three-dimensional structural characteristics of the proteins and ligands, we used 8 GPUs, and just to do 100 epochs, it took us 32 hours. The reward sums, for example, we just used a single GPU, and it took us a couple of hours to do, and that was 30 times faster than the time it would have taken if we were to just run that on CPUs. The generation of 300,000 molecules took about 3 hours on a single GPU, and that was 10 times faster than the equivalent CPU. Another key ingredient to our success was a small, very agile team of superheroes, and it's a privilege and an honor to work with them. Here's their actual faces. So Ashwin Rathod is our VP of Innovation and Data Science. He understands AI, and he has a very impressive background and entrepreneurial spirit, and he really is a go-getter. Vivek Singh, a senior data scientist, he's also a biologist, but specializes in bioinformatics, and he was really the center of and the heart of this effort. So we really appreciate all the work from Vivek. Bibash was outstanding. He really understands how to use machine learning to solve complex problems. He pretty much wrote the majority of the code, and we are thrilled to work with Bibash. And Rohit Yadav was also outstanding in helping us validate our work and keeping us honest. With that said, this is our card. It would be great if you like to reach out. Would love to hear from you, and we are ready to answer some questions.